Hello everybody, it's time for Enzymes, the best module in biochemistry, in my super biased opinion. So after watching this video, you should be able to define key vocab terms, identify substrates, products, and enzymes in a chemical equation, describe how enzymes catalyze reactions based on formation of ES due to selective substrate binding in the right orientation and proximity due to electronic, stereochem, and geometric complementary. The preferential binding of the transition state and the overall effect of both of these on a reaction coordinate diagram. Write and interpret a reaction mechanism for a general enzyme catalyzed reaction and list the different types of classifications. Okay, I have no idea if I said let's get started too many times in a row here. I had to kind of splice some of this together. All right, so here is the hydrolysis of a peptide bond. So there's a peptide bond, you remove water and you get the two pieces back out, the peptide and in this case the C-terminal amino acid. And it turns out that this takes years without a catalyst. Okay, this reaction would take years, and we know it doesn't take years because when you eat a protein and it goes in your belly, it doesn't sit there for years and years and years. Also, just food for thought, at what pH is this uncharged and this? This one, it would have to be above pH 10. This one would have to be below pH 2, so just food for thought, this is not possible. This structure is not completely accurate. Anyways, but this reaction, peptide bond hydrolysis or breaking of the peptide bond, takes years. How do biological reactions proceed on a reasonable time scale? So how do biological reactions proceed on a reasonable time scale? You have two options for increasing the rate of reaction in a biological context. One is you can increase temperature. Not very reasonable, because at certain temperatures, when we get too high, proteins fall apart, DNA falls apart, RNA falls apart. So that leaves us with option 2. We got to add a catalyst. Okay, and in this case, our catalyst is going to are called enzymes. Okay, so enzymes are biological catalysts. We're just going to do a couple things here. Catalyst means that they speed up reaction rate without being consumed. They also do not alter the delta G of the reaction. So enzymes don't change the energy of the reactants or the energy of the products. They just get from reactants to products faster. Most are globular proteins. Some are not. Some are embedded in a membrane. Some are also made out of RNA. So for example, the ribosome is an example of an RNA-based catalyst. Food for thought. And then they catalyze the transformation of a substrate. This is a substrate. These are the same thing, to the product. So the substrate or substrates can be plural, are the molecules being chemically altered. These are different from ligands, so ligands also bind to a protein but are not chemically altered. So oxygen is the ligand for hemoglobin. When it comes back out of hemoglobin, it's still oxygen. Here, maltose, the substrate, goes in, and when it comes out, it's glucose, so it's been chemically altered. So the substrates are here, products are here. You usually put the enzyme somewhere near, the enzyme name somewhere near the arrow. So here, Here's the enzyme name. Here's the enzyme itself. Hopefully, maybe, you can see that the substrate is right here. So the substrates bind to the enzyme, are converted to product, and the products then leave the enzyme. Okay? Just a little bit more basics before we get into specificity and binding. So enzymes are typically named for one of two things. One, the reaction that they catalyze, or two, catalyze, or two, they can also be named for their substrates. Like as in maltose is named for its substrate, not what it does. Because the substrate was, maltase was named for its substrate, maltose. All right, so here's some 6 main classifications of enzymes and what they do. So oxidoreductases, those catalyze redox reactions. Okay, so movements of electrons. A transferase catalyzes the transfer, so I'm going to put X for as transfer of functional groups. So this might be moving a methyl group from one substrate to the other substrate. Hydrolysis catalyze hydrolysis reactions. Okay. Lyases catalyze breaking of bonds, not through hydrolysis. Isomerases, hopefully that sounds like what it does. It catalyzes an isomerization. And a ligase catalyzes the joining. Go on back. Joining of two substrates. Substrates are equivalent to reactants, if that's an easier way for you to remember them. So in biochemistry, we're going to use what I call a general enzyme reaction mechanism or equation. And you might want to commit this to memory. We're going to bring it up over and over and over again, so it would be a really good thing to remember. So if you remember for KD, we had protein plus ligand equilibrium to PL. This is sort of similar to that. So a general one is the enzyme and the substrate are free. There's a binding event where they come together, but yet nothing catalytically has happened. The substrate is not yet converted. Again, we have a little k here for the rates, an on rate and an off rate. Okay, so this first part right here is just binding, which is facilitated by specificity. Not bad. Okay, and then the second part is catalysis. So this is where the substrate is being chemically altered, bonds are broken, bonds are made, something is changing in the substrate to a product, and then that product leaves the enzyme. So in this case, the second part right here is catalysis. Okay, and we call the rate here with a little cat for catalysis. That rate is for catalysis. So the first part is binding, the second part is catalysis. One other thing that we should know, in this most simplified version, we are assuming that product does not rebind to enzyme. That's why we have a single-headed arrow in the second part of this, is that we're assuming that product does not rebind. Okay, so today's video we're going to focus on just this first part, and then videos later will focus on catalysis. So again, today is just about binding and specificity and the thermodynamics of that. It's not about catalysis. That will come later. Okay, So what I wanted to show you here is this really beautiful image that I actually got off of Wikipedia. If you look at the enzymes page for Wikipedia, it's pretty legit. They did a really nice job, and they've got a lot of really beautiful figures. So what we've got here is this is lysozyme, and it is binding to its peptidoglycan substrate. So this is the substrate. NAG and NAM, if you're in microbiology or have taken microbiology, NAG and NAM are part of the peptidoglycan layer, okay? So its substrate here is fit right in this pocket or cave. So if you'll notice, there's kind of like this cavernous area right here, and that whole area with the blue and the red is called the active site. Okay, it's where the substrate binds, and the binding residues are in blue. It's also where catalysis will happen, but those tend to be different residues. So the binding residues or binding amino acids that are in blue here are not always the same as the red ones. They can be, but they're typically different. So the active site is where binding happens and catalysis. So again, today we're only going to be talking about binding, and next time we'll talk about catalysis. And what I want you to see here is that a really good chunk of this protein is just scaffolding. It's just for structural support to make the giant cave that is the active site. So lysozyme, again, in this chemical equation is sitting on the arrow. The products are here. It catalyzes the cleavage of that glycosidic bond. So just so you know, the active site, that's where, right here, I don't know how many different arrows I'm going to draw to it. It's like a cave that the substrate goes into in a specific manner. The substrate is then converted to a product and then released. The quote-unquote how do enzymes work video is so awesome, and it does a very beautiful job of showing this. Hopefully you'll watch it after this video. It might make some more sense, but watch it as many times as you can to pick up as much as you possibly can. Okay, so how do enzymes increase the rate of their reaction? So this is going to be a two-part story, but essentially, well, maybe I don't want to say, oh, hold on, let me back up just a little bit. Okay, how do enzymes increase the rate of the reaction? The most basic answer is that they decrease the activation energy. Okay, and if you don't remember, the activation energy is the bump there, okay? We don't need to see that. Okay, in order to do this, we have two parts here. There's two things that it does. And the one thing is that it binds substrate with high specificity. You don't want every enzyme being able to bind to every little small molecule that it binds to, that it comes in contact with. That would be completely inefficient. If an enzyme needs to bind only to glucose, then only glucose should fit and stay in the enzyme active site long enough to be converted to product. You don't want that glucose going into lysozyme. You don't want that glucose going into maltase, so on and so forth. So it should be bind with high specificity. And then two, so it's going to select its substrate with high specificity. And then it turns out, and this is going to sound really, really weird, but hopefully we'll get to a good example of this. It turns out that the enzyme, let's say it this way, preferentially binds the transition state and lowers its energy. Okay, so we're going to talk about specificity first, and then we're going to talk about binding. The transition state, again, this is the generalized enzyme catalyzed reaction mechanism, and we're doing bind. Okay, before we get into specificity, I just wanted to give a shout out back again to our good friend, guess what this is? Ordered water molecules, disordered water molecules, the hydrophobic effect. Okay, So here the entropy of the water is favorable. You see the substrate is surrounded by a lot of water molecules. The enzyme is also surrounded by a lot of water molecules. When these two interact via three different things, but one that is shown here are the non-covalent interactions, hydrophobic, hydrogen bonding, ionic, and so forth, we free up a whole bunch of water molecules. So the binding of substrate to enzyme is driven by the hydrophobic effect. And you get to free the water, free the water, free the water. But the hydrophobic effect says nothing about specificity. Go there next. Okay, selective binding and substrates. There are three parts to this, and you have to have all three parts in order for this to work. The first one is stereochemistry. Okay. The second one is electronics. These are our charge-charge interactions. And then the third one is shape, also known as geometric. Complementary. Complementary. There we go. I really like this figure right here. We're actually going to come back to it, but it shows shape and electronics very well. It actually doesn't quite show stereochemistry very well, but let's go in order. Stereochem, electronics, and shape. All right, so in order for an enzyme to bind with high specificity, they select for a single enantiomer. And how that's done is via the amino acids in the active site. I really like this figure. So remember, enantiomers are mirror images. So here's the chiral carbon right here. It's got 4 different things coming off of it. In this case, a cube, a diamond, a sphere, and a pentagon. Okay, and they're oriented differently. They're mirror images. The enzyme active site is down here. If you notice, it's the same between the two. The active site is not changing. But we do have 2 enantiomers, and only this enantiomer will bind because it will box to box, diamond to diamond. Okay, that's a pyramid or a triangle. Dang it, it looked like a diamond. And sphere to sphere. Okay, so this enzyme selects only for this enantiomer. It will not, the sphere will not fit here, the cube will not fit here. So stereochemistry is really important because a lot of our molecules, our substrates, are chiral. The enzyme itself is also chiral. Ooh, food for thought, weirdness. But stereochemistry is one of the 3 pillars of selectivity. The next one is electronics. I'm hoping this one is the easiest for you to grasp because this is the non-covalent interactions. Okay, so this are the charge-charge interactions. Charge-charge meaning charge of the substrate and charge of the enzyme. Charge-charge interactions between the substrate and the enzyme, or more specifically, the amino acids in the active site. Okay, so I'll walk you through this image. So you're going to start over here. So this is only showing primary structure right now. There's no secondary structure, and what they've done is they've made all the amino acids that are going to be in the active site these little Christmas looking lights, okay? And when the protein folds, all of the Christmas lights are in the active site. So what I want you to first see is that these amino acids in the active site are actually far apart in sequence. Like some of them are close, but most of them are far, but they end up being very, very close to each other in space, okay? And what this does is it creates certain side chains sticking into the active site, and the substrate is going to bind with high specificity, creating specific non-covalent interactions. So this right here is the substrate. And when it gets into the active site, the amino acids in the active site, which are what were all the Christmas lights, and they're all in colors here. They're going to select for particular stereochemistry, like you saw before, and it's also going to stabilize the interaction between the enzyme and the substrate through all these non-covalent interactions. Look, there's a salt bridge. Let's see, all of these are hydrogen bonds, and it looks like all of these are also hydrogen bonds. So electronics stabilize. Also, more favorable bonds that we make, the lower the delta H, which translates to a lower delta G. So that is stabilizing when we have lots and lots of non-covalent interactions. That is energetically stabilizing. And the third piece of the puzzle is geometric complementary. So this means that the puzzle pieces fit together. The shape of the substrate will fit into the shape or the absence of space, the cave of the active site, which again is dependent on the active site amino acids. Okay, so what I like about this is that it shows that this substrate shape-wise will fit into this active site, so that's geometric complementary. It will also show that once this slides in here, the negative charge here will have a favorable interaction with this positive charge. Some amino acid, maybe it's arginine, maybe it's lysine. You've got a hydrogen bond right here, and these H's are hydrophobic effect. So these are all the non-polar pieces. So you might imagine that that's a serine. That could be a serine. This could be a valine. This could be an isoleucine. That could be a leucine, so on and so forth. Oh, look, here's another hydrogen bond. Another hydrogen bond. Who knows? That could be a threonine or a tyrosine or another serine. So the three pillars of the selectivity are shape, geometric complementary, electronics, non-covalent interactions, and stereochemistry. Now, what I've described so far is what you would consider the lock and the key, where the key fits specifically, the key is the substrate, fits into the lock, which is the enzyme, and nothing ever moves. But it turns out a lot of enzymes actually have what's called induced fit. Okay, that's where after substrate binds, the enzyme changes shape to better bind the substrate. Okay, S here is substrate. So this enzyme right here is hexokinase. It binds two things as its substrates. One of them is glucose and one of them is ATP. You can see them as these two different shapes. And then after they bind, the enzyme changes shape, so it actually kind of closes around them in order to better bind them. So that's an example of induced fit. You would say that hexokinase has allosterics. It has two different shapes. This is one shape, this is a different shape. Food for thought. Okay. So far, I just kind of want to tell you where we are before we go into part 2. So far on a reaction coordinate diagram, we have our substrates here, our products here, and you'll notice that the solid line is with enzyme, and the dotted line is without, okay? So you have a really, really high transition state without the enzyme, and then with the enzyme, what you're looking at is two parts here. One is the binding of E to S, and then the next one is the lowering of the transition state itself. So it's actually the culmination, if we were to average these, that causes the overall decrease in transition state energy. So it's not just that this piece is lower, but this transition state energy is able to be lower because we form this ES. And ES is enzyme-substrate are bound together with high specificity, okay, that is facilitated through stereochem, electronics, oh, yikes, come on, pen, stereochemistry, this is a parentheses, stereochemistry, okay, I'm just going to rewrite them, yep. It's not going to let me go back that far. Where is my eraser? There it is. Okay. High specificity via stereochemistry, electronics, and shape. Okay? So the overall decrease in transition state energy is first facilitated by binding the substrate to the enzyme. That's actually very low in energy. These values are called intermediates. Okay? It's very low in energy because of all the non-covalent interactions. Okay? So again, our two-part story, an enzyme, in order to be a catalyst here, has to lower the transition state energy. That means this bump has to come down. And there are two things contributing to this, and you must have both. You must form the ES, okay? And then when we move in our reaction coordinate, when we move towards P, it's going to turn out that when the enzyme, when we start to move into the transition state, so we've got partial bond breaking, partial bond making, that is actually going to bind to the enzyme preferentially. So let's just get to that so I can stop talking. All right, so I love this example. So this is an example, this is part 2, enzymes preferentially bind the transition state. And this is a fake enzyme. It's called stickase. Stick and literally stickase, all right? So here's the uncatalyzed reaction. You have your substrate, you have your product. It's thermodynamically favorable because our delta G is negative. And then we have our transition, all right? So our substrate is a metal stick. Our transition state is up here green. That is the bent stick. That is the highest in energy. You can imagine physically grabbing onto both ends of the substrate and starting to push on it to get it to break. The bent is the highest in energy, and then you have two products and they're broken sticks, and those again are lower in energy. Now, if we add an enzyme, and this is the enzyme right here, this blue bubble that they call stickase. Hilarious, right? Because it's an enzyme and it breaks a stick, so it's named after its substrate. You get it? Cute. I think it's cute. There's two options here. Now, the first option right here is that enzyme binds S preferentially. Okay, enzyme binds S preferentially, so that means that the active site right here is shaped only for substrate. And what that would look like is that the ES is super low in energy, but the transition state is still just as high. I mean, it may have come down just a little bit, but it's still just as high, so you do not gain, the enzyme gains nothing by binding the substrate best. It binds it well with selectivity, but it doesn't bind it best. Now here's the example where stickase, this is your option 2, where the enzyme binds the transition state preferentially. So the enzyme binds the substrate and forms ES, and there are non-covalent interactions, and there is specificity here because it does fit the shape. And obviously we have no stereochemistry here because it's a stick. Okay, as we move to the transition state, look at that. The enzyme is shaped not only to bind the transition state preferentially, but look at all these extra non-covalent interactions. So you have shape and electronics are best between enzyme and transition state. So what this translates to here is that step number 1, the formation of the ES, is stable. We do get a lowering. We do get an intermediate. Step 2, creating the transition state, is also lower in energy. See how much the bump went down? And then step 3 here is the products are released. So you see them here. So this is my example. This is the best example I've ever been able to see that cartoon-wise shows you how enzyme binds to transition state preferentially. And then again, you get the overall average lowering of the transition state energy because you not only formed ES that was stable, but the enzyme stabilized the transition state. Okay, in order to do this, I've got to talk about 2 more things that we've actually already covered on in this class, but I have to say them again because they're still important. The orientation of the substrates is best, I'll say, in the transition state. Okay, so the orientation of the substrates is best in the transition state. And so what you see here is you've got one substrate that's got this curve, and you've got a second one that's an arrow, and the enzyme should bring these together so that this curved part is near that curved part. That's orientation. So they're brought together in the right orientation so that you can actually get a reaction between the two. What you don't want is the enzyme to bring them in so that it looks like that. That would be bad orientation. Okay? And orientation is best during the transition state. And this again is facilitated by the amino acids in the active site. Okay, so that's orientation. And then proximity means that when the substrate or substrates enter the active site, they are, 1, close to each other, and 2, fill up the available space. Okay, and again, orientation is best in the transition state. So here you have the 2 substrates. When they bind with the right orientation, they should also be in close proximity, so they should be near each other. And then here in the transition state where the bonds are starting to be broken and starting to be made, you have the strongest binding and the best proximity and the best orientation. Okay, so here's our summary up to now. This is the reaction progress. So this is substrate, this is product, and the reaction progress here goes, this is E plus S, goes to ES, which is, oh, maybe I should get this all nice and lined up for you. Kind of hard to do, but let me try again. Okay, so here you have E plus S in equilibrium with ES. This is ES right here. And then to product to here, and in the transition between ES to product or S to product, you have ES transition state. Okay? This lowering of energy, that valley, that intermediate, is facilitated by the decrease in energy of binding the ES. This is due to electronics primarily, but this also describes specificity in terms of stereochemistry and shape. Okay? The lowering of this is because the enzyme bound the transition state, didn't say better or best, better than the substrate in optimal orientation and proximity. And again, the lowering of this The lowering of this is due to primarily a decrease in delta H from all of the non-covalent interactions between transition state and enzyme. And that's all I have for you. Again, watch the thermodynamics videos, all 3 of them. Watch this video, watch the How Enzymes Works from PDB 101, and I will see you in class. Bye-bye.